This is a little bit the outline, but in general, yes, about clinical studies, what to do or maybe not to do. In fact, today, when we go to meetings, everybody always talks about evidence-based medicine and how we should do that. And when you talk about evidence-based medicine, a randomized controlled trial is basically considered a type of investigation that you should do if you want to do a study in a scientifically correct way. This is, however, easier said than done, because if you want to do a randomized controlled trial in a proper way, it involves many different aspects, and I will go through some of them. And it's not always possible or maybe not even necessary to really perform a randomized controlled trial. As an example, recently I heard somebody making a statement like, let's say, ICSI. Never any randomized controlled trial was done. So should we stop it and go back and do it again? I don't think so. But it wasn't done, but that was a different time. So you can also ask, like, is an RCT the only way to answer a question? And when you evaluate a method or a product, when is it really a study? Because a lot of things that you do in terms of quality management system are validations, and a validation is not the same as a study. I listed a few names here, and this depends on, let's say, which part of the world you are in, because if you read documents of FDA, they talk about trials. If you read documents of EU, they talk about clinical investigations. So that's another thing. You have different words to determine more or less the same thing. Certainly in Western Europe, quality systems have been introduced during recent years. Not yet in all parts of the world people are that advanced. And when you have a quality management system, then of course a validation is part of that and is becoming part of your daily routine. However, validation is not the same as doing a scientific study. And what is very obvious is that the number of cycles that you do in the clinic where you work really affects how you can do validations or how you can do studies. If you are looking for some guidance, if you want to do a study, well, for a validation, as said, it's part of your quality management systems, and these are the most commonly used when it comes to the IVF laboratories. When it comes for clinical investigations, there is an ISO standard that nicely explains basically how you should run a clinical trial or a clinical investigation. There is also the Helsinki Declaration on Ethical Aspects, if you want to see what should be done, if you want to do it in the right way. And of course, very important points here, if you think about studies, are how many cycles, how many patients, how many embryos do I need to include? And again, for a validation, you have your quality handbook that more or less determines this. But if you think about a randomized controlled trial, then you need a power calculation to help you determining how many cases you should include in your trial. Now, a few more words about evidence-based medicine, and this is also, in fact, something that has come up during the last 20 years, more or less. And this is a definition that is quite often referred to in a paper from '96. And when talking about evidence, there are different levels. Of course, all starts with a single case. Sometimes you see case reports where in literature people publish about one single event, where usually they try to find a combination of treatments that has not been published yet. And the next level is, in fact, a case series, where you do a number of treatments, you do not compare, you just report, we did so many patients with treatments X, and this is our observation. Then you have like uncontrolled or observational trials. These can be either retrospective, prospective, but they can be done in different ways, but in practice it means that they are not really controlled for. This may be, let's say, from July to December you use medium A, from January to June you use medium B, and you compare the results. This may be very valuable for you, but from a scientific perspective it's not the right way to compare methods or products. So a randomized control trial is the correct way and the best level of evidence to try to prove that a method or a product is better. And then the ultimate level of evidence is a meta-analysis. So usually after a number of publications, somebody is interested and does a meta-analysis and publishes that. And it's quite interesting. We had an internal little discussion about this. If you take time-lapse and you look it up in PubMed, you get 125 titles, more or less, and 29 of them are reviews. So people are quite interested, apparently, in doing reviews and meta-analysis on things, maybe more than doing the actual studies, which are maybe more important. In daily practice, do we really support evidence-based medicine? And this is interesting when you talk to people, visit clinics, ask for opinions. Sometimes you have people say, no, this is best. This is what I do. So this is maybe not the way to progress, because in some way this is a dogma. No, I only believe in this and this is what I do. Another comment that is also quite common is we always did it this way. So why do things differently? So that is not something that allows you to do evidence-based medicine because you adhere to tradition. Another comment is everybody does it this way. And that is quite interesting because everybody often stops at the borders of the country. And at the next country say everybody does it this way, but a very different way compared to the other country. But also that is not probably the best way to have evidence. So it's not that often that people, when you talk to people, they say, no, I do this because the recent literature told me that this is probably better. But in practice, this is what we should adhere to if we really want to progress. You could also ask how much should we adhere to it, because as already mentioned, the number of cycles you do really affects how easy it is to really compare products or methods. And another aspect is that findings from a study are not necessarily reproducible. Because this you know very well, there are so many differences, so many parameters that can affect the findings from any study that you do. And I just show you one example that I found from recent literature. This is a paper published earlier this year. They compared different volumes for culture in 2 steps, and the lower volume being 7 microliter droplets, saying that they found higher blastocyst rates using this small volume. In the same journal, this paper is now in press. Reduced blastocyst formation. They used exactly the same volume and a small volume, and so they have opposite findings. And this shows that that not necessarily everything you do is reproducible. So that also shows the importance of a meta-analysis, because it puts together all similar studies and then compares whether a method is better than another one. So what is maybe worth talking about for a few minutes is like, when is something a validation or when is it a study? And of course in your quality management handbook you define your validations, and it describes when, how many, how you should do things. And again, this is really affected by the number of cycles that you do by your policies. Because the number of cycles you do really affects how many cases you can include, being it either embryos or patients. Nevertheless, there are always some statistical considerations you should keep in mind, but the validation is quite different from a clinical study. Now, if you really would like to go that way and perform a study in a scientifically correct way, there are again these levels of evidence you can think about, and as said, the randomized controlled trial is the best level to work with. Now, there are a number of aspects to look into, and a very important one you should ask very early on: what is the study question? Because basically, if you want to compare method A with method B or product X with product Z, you can have one question with one answer, and that's your primary endpoint. Sometimes you read in papers, we want to know whether this procedure had an impact on A, B, C, D. That's not really possible. You can only analyze the impact on one outcome parameter, and you power the study for that, and all the other endpoints are in fact underpowered. I don't say they are not important, not relevant, that's another thing, but statistically they will be underpowered. Only your primary endpoint will have solid statistical power. Another discussion is how you compare groups. Certainly when you talk with clinicians, they will always say that using the patient is the only right way to compare methods. I think for the lab, using oocytes or embryos as the unit can be very useful as well, certainly also in validations. Let's say you want to compare survival and slow freezing vitrification. You cannot necessarily have an implantation or live birth as an endpoint because you want to know how survival is, for instance. So different reasons why you would have different endpoints. And then a lot of work and a lot of time is in fact your study protocol. This is not something that you write on half a page. It's a lot of things that need to be part of that. And if you look in that ISO standard, you can see a lot of examples on how these documents should be developed. It's a lot on the objectives and the hypothesis, your study question. Design has a lot of aspects. One, for instance, I put it separate here, but it belongs under design, is reasons for termination. You should define from the beginning if you would stop the study, for what reason you would stop the study. So a study protocol is something that gives you a lot of work. And of course, it's not just performing the study, doing the cases, it's a lot of additional paperwork. You should in fact have an information leaflet for the patient. Of course, a consent form, either if you would compare a approved product, if you do it as a randomized controlled trial, the patient should sign that she or that they accept being part of that study. You have to have your ethics committee application, and depending on the country or regulation, maybe also an authorization of a national body. And if you want to publish it, it should also be registered in a public database. If you go on the journal's websites, they will tell you which databases are accepted. And then, of course, a lot of paperwork is in the data registration and the reporting. As I already touched on, the power calculation is a very important part of your study design and should be done quite early on, because this really tells you how many cases you need in the study you want to do. Because you want to know how many you want to have that any significant finding is not by chance. And as already mentioned, most studies have secondary endpoints, and as I said, they can be very valuable, very important, but from a statistical point of view, they are underpowered. And this you should do quite early on, so that, because this really affects the study, how you do it. A few more words about power calculation. In fact, you can have different study designs. You can have a superiority trial, so you want to see whether product A gives you better results compared to product B. You can have an equivalence trial or a non-inferiority trial. Just some examples of a power calculation. Let's say you have a pregnancy rate of 32% and you want to do a prospective study, and you say, okay, if I have a difference of 5% more for product A, I'm happy. So if you would like to do a study like that, you will need to include 2,800 patients or 1,400 in each group. If you say this is a wonderful product, it will give me 15% more pregnancies, then you would need only 326 in total. So you see, it really depends on the difference, how many cases you need. Let's say you have a 55% blastocyst formation, you want to compare with another product, and you are happy with 5% more, you would need 20,000. So quite difficult to do. If you say, oh, this will be 15% better, then only 500. So just to give you an idea, if you want to do a study in the right way, it really is important to do this, but also complicates to do studies in the correct way. When reading papers, sometimes you get the impression that by equivalence trials or non-inferiority trials, people kind of state, then you need fewer cases. And therefore, similar examples, it's not necessarily the case. Because for an equivalence trial, you say, I want to prove that A is as good or is identical to B. Here you have to define the range that you accept, and then you see you still need quite a large number of cases, not necessarily much less compared to a superiority trial. Also the same for a non-inferiority trial, where you say A is not worse than B. You can take the same percentage for both groups, or you could say, okay, with B I have 32, with A I'm happy if it's 28. That gives you different numbers, but if you would take again 32% for both groups, these are the numbers that you would need to include. So just as an example to show you that if from a statistical point of view you want to do a study in the right way, it really requires large numbers. Another important aspect in an RCT is the randomization, because you want to have a correct distribution. And for the lab, this is not very practical. We're very much aware of that, because it really affects your workload, or the way you organize your work, and it requires a lot more attention compared to routine work, where you do the same for all patients. There are different ways how you can do a randomization. That also affects if it's a single-center trial or if you are having a multi-center trial. You can do a block randomization, so different ways. Practically also, you can, let's say, the more old-fashioned way is using sealed envelopes. Today, people use more and more electronic lists. But then you should also make the list in a way that you cannot, let's say, skip certain patients, that you go only to the next one. And very important is your point of inclusion. For instance, I remember the discussion we had in Brussels at the time that we were comparing day 3 with day 5 transfer. Do you include the patients at the time of counseling before the treatment? Do you randomize only, for instance, on day 3, depending on the number of embryos? So these are very important points to discuss very well on beforehand, because it can give you a very different picture. And then also very important, blinding. That's not always possible, but depending on how you're organized in the lab, this may be possible. Let's say you compare 2 methods of vitrification. The person doing the warming has to be aware of the method, because that person needs to use the right protocol. But if a colleague is doing the embryo evaluation, that person may be blinded for the embryo assessments. So all different things to look at. A few more words about primary endpoint. So this is really important. And in fact, personally, I think in IVF, we are in some way biased, because usually you have more than one oocyte, very often more than one embryo, and then always some kind of selection is involved. If you have a study where pregnancy or live birth is the primary endpoint, we always have some selection. So, and that makes it, I think, more complicated to compare the true value of method A and method B. And when discussing with people, the embryology lab or the embryo lab parameters are not always really considered. As I mentioned, sibling oocytes can be quite valuable sometimes, but not everybody considers that as good as a study. Other parameters are maybe not really easy to compare. Live birth as an endpoint, again referring to survival after slow freezing, for instance. This is my personal opinion. I think if we want to really compare how method A and B works, that we should be able to include all data from a cycle, not having just the transfer in the fresh cycle, but also the cryo. Because that's the only way to ultimately compare what does a cycle give you, one method versus another method. But this is very unpractical because that takes several years to complete such a study. Some things that we still observe in studies and should maybe not done. Quite frequently you still read the papers that have not included a power calculation or say that for some reasons they have no ethics approval. Strictly spoken, you should have that. In terms of protocol, still a lot of things where we feel this can be better in an exclusion criteria definition, information on reasons to stop the study and other aspects as well. Or that the randomization is not very clearly described. Statistics not always really appropriate. And sometimes people tell you that, oh, we did this pilot study, the result was so obvious, we stopped and we do this. So a pilot study can be very valuable, but it's not really the final evidence. In summary, as I started with today, a randomized controlled trial is considered the golden standard, but not always easy or possible. And it takes you a lot of work if you really would like to run a clinical study in the right way. There are guidelines available that can help you in preparing it and performing it. And I think that sometimes there is a mix-up between validation and study. And personally, I think that it should also be accepted to present validations as, for instance, an abstract at a scientific meeting, because it can be a good number of cases and can still be quite valuable. And this is very important and not always done correctly, the power calculation and the randomization. And since you sometimes need large numbers to do a study in a correct way, I think it can be quite useful if you can collaborate with different clinics because it make it easier to run those studies where you need large numbers. Thank you.